Welcome to BioIT World's Trends from the Trenches podcast, your insider's look at the science, technology, and executive trends driving the life sciences. I'm Allison Proffitt, editor of BioIT World. This episode is hosted by Stan Gloss, founder of BioTeam, a life sciences IT consulting company at the intersection of science, data, and technology. Joining Stan today is Terry Lowe, CEO of Vizgen. The two talk about spatial biology, why it matters, and how Lowe sees the technology changing healthcare's future. Let's listen in.
Good afternoon, Terry. It's so nice to be talking to you today. I've actually had this podcast circled because I'm fascinated by your website and the concept of spatial genomics. I've been involved in genomics for the past 20 years with the first DNA sequencer that I ran into called the Celexa machine before it was Illumina. Mm-hmm. So maybe we can start it off by having you tell a little bit about yourself and a little bit about your background and how you got to Vizgen.
Yeah, first of all, Stan, it's great to be here. I really appreciate the opportunity to talk about spatial biology and spatial genomics, and happy to give a little bit of my own background to kick it off. So I've been in the healthcare space for almost 25 years now, mostly in business and commercial type roles. I've covered the gamut from pharma companies to diagnostics to life sciences. But the last 7 years has really been focused in this particular area of spatial biology. And I'll have to say, I've been lucky enough in my career where I've been involved with many different types of new technologies and breakthrough products. And this has got to be, without a doubt, the most exciting space that I've been involved with. It's really been pretty amazing to see all of the enthusiasm and momentum that we've gained in this spatial biology field. So I started focusing on spatial biology actually when I was at PerkinElmer. I ran the tissue business there. And then we later spun that out to become part of Akoya Biosciences, where we created a fully dedicated company around spatial biology. And then most recently have come over to Vizgen now as CEO for the past 2 years.
So maybe you can help us and our listeners about what is spatial biology and spatial genomics? And then maybe follow it up by telling us, why should we be interested in this?
Absolutely. So I think maybe one easy way to think about it is really taking it from a technology perspective. And you can almost think about it as bringing together two very different scientific approaches where we're now able to combine and get the most valuable information. So on the one hand, you've got what is really has been historically a very visual type of approach where you're looking at sort of macro level types of information. And that has come out through the histology side or histopathology. And then on the other hand, you have this genomic side of things, or even beyond genomics more broadly, to say that we have the ability to look at molecular markers and molecular markers at a very, very high level or very high plexing. So the challenge has always been that these two have been very separate types of fields of science and technology. And now we have this ability to bring them together. And that's really how you get spatial biology, because now you have the ability to take both the visual side or understanding kind of understanding the information at these different scales of biology that you get with histology. But then you have the tons of information that you get from the molecular side, where you have all of this high parameter data that you can now build into what you're seeing visually.
That's terrific. It's interesting because we have lots of conversations with our clients about this idea of multimodal analysis and people wanting to mix different things together to be able to derive new viewing or understandings of the data that they're looking at. So it's just interesting to hear how you've recently combined that And so, again, what will this do for the scientist that's listening to this, and or the drug discovery company who really is trying to accelerate their time to discovery?
Yeah, so I think one of the challenges that we've seen in the overall life science research field is really this ability to understand within a biological system what is happening. So the genomics approach has really been to kind of get to a very, very detailed level of molecular information where we understand the starting code of where the biology really starts. On the histology side, you're kind of seeing the end result of what actually is happening there. And so what we've kind of missed, by not being able to put these together is really understanding how some of these specific mechanisms, specific molecular interactions lead to these changes in the biology, lead to what's happening and why disease progresses, what is generating those changes. And so by using this genomics information that we can now collect, but understanding where that information is located, within specific cells. And so this allows us to have information about specific cells that are in the tissue, where those cells are located, what those cells are, what those cells are and what those cells are doing, and how they're potentially interacting with other cells within the same space. So we're actually able to see now that there's different types of cells that are collected together, and let's call them certain neighborhoods, where they have a certain way of interacting with each other and they have certain functions. And each cell has a different role to play within those neighborhoods. And that's what we're kind of learning now with this molecular information that we have in spatial context.
Mm-hmm. That's a beautiful metaphor, because I was trying to think about a recent interview I did with Recursion Pharmaceutical, and it seems like they're trying to understand. They do many different experiments, and they take normal cells and perturb them, and then image them in some way to see, trying to decode the biology. But you use the word neighborhood, which now implies, instead of thinking about tissue as this is the house, you're actually thinking about a number of houses in the area and how tissue kind of works in a neighborhood of other cells. For instance, you could imagine somebody having a diagnosis of a tumor, and then starting to say, because we're always trying to excise out the edge and make sure that we've captured everything out when we remove a tumor. So we could easily imagine a neighborhood where you have good neighbors and bad neighbors.
Yeah, absolutely. And I think what's interesting is when you're able to do this type of cell typing, you can certainly find who your bad neighbors are. But sometimes it's also understanding, a neighbor in one environment, may act in a certain way and act in a different way in a different environment. So just because, it's the same neighbor or the same character or the same cell, the way that they function, the way that they act can change dramatically depending on the environment that they're in. And that environment really is influenced by the other cells that are around them, the other, external environmental activities that are happening around them. And that is how we change the way that cells respond and how they act. So you have, what we generally classify as different cell types, where, we can say that this is a T cell or B cell or tumor cell, but we also have cell states in genomics or in gene expression, where they're acting in a certain way, even though they are one type of cell. And so at the genomics level, this is the level of detail that now we can see with the gene expression, why this particular cell may be acting in a certain way that we couldn't really identify before. And it really is related to their spatial context of who's around them and how they're interacting with them.
So is the fundamental idea, if you understand these cells and how you put them into certain neighborhoods and they act a certain way, either upregulating or downregulating, a reaction is the idea that mapping those into kind of an encyclopedia of these cells act a certain way in certain environments allows you to kind of tease out some of the systems biology. Is that what you're trying to ultimately achieve?
That's exactly right. And I think the concept of cell or tissue mapping is a very good description. That is a concept that actually started with single cell analysis, with single cell genomics, where they tried to identify all of these individual cells and what types of genes were being expressed. So there's a lot of initiatives that have started already over the last few years to do this type of tissue mapping. What's been a challenge with that approach, though, is that with single cell genomics, you're really disassociating that tissue so that you've got these individual cells. And so what you end up with, instead of having an atlas or a map, is really more like a dictionary, right? You've got your cell, you've got the definition of what that cell is, but you don't really know how it is in context, right? You'd rather read that in a book, or you'd rather see it in a picture so you can see how these terms or how these cells can be applied. And that's what you get with spatial. You don't just get a definition of what that cell is. You get to see where that cell is located and how it's interacting with other cells. And that is how we understand, or how we can better understand, the systems that relate to what's happening in the biology.
Yeah, it's really interesting because I've been doing some reading about in the business side with a guy named Clay Christensen. He talks about jobs to be done. And he talks about things that are, understanding whether or not something is correlating or versus is it in context, and what's the context. And he's trying to help people understand how to look at things, especially when you're doing system level thinking, how to see it's so important to see it in context.
Yeah. And one of the things I think if I go back to my sort of description of how these have been sort of the convergence of these two different approaches, I mean, we've had the benefit of histology, where we can see how things are organized, how we can understand how the tissue architecture works, but we just don't have as much information about the details within that to really understand what's happening there. And, one of the things that's quite different on the genomics side is that information, whereas you can't see it visually, genomics has been great in the sense that you have all of that information which has been digitized. So you're able to do very, very high level parameter analysis computationally. Whereas, if you think about a histopathologist who's looking at a tissue under a microscope, you can't really absorb so many different parameters simultaneously and understand how these are all interworking together. So one of the great things about what spatial does by bringing these two things together is that it's really the ability to computationally now analyze the information that you have, because you have so many different parameters that you have to tackle. And this is really one of the important advancements of how you're getting the best out of these two approaches. And what we're seeing now is that this concept of tissue mapping is really taken off, across a lot of research areas. And a great example of that is in the Brain Initiative and other initiatives around neurosciences, where as a starting point, what we're trying to do is we're trying to map the brain. We're trying to figure out how the brain works by actually creating a physical map of where all those cells are located and what those cells are. And this, has been a process that, again, started with single cell analysis, but now has moved into the spatial biology world, where we can actually create these actual maps of what the brain looks like. And this is the starting point, really the foundation of where, as we figure out how different diseases potentially will progress, you want to start with the healthy tissue first, right? You want to start with a healthy brain to see, hey, here's what it looks like when it's normal. And then I can now look at, these other conditions where you might have a diseased brain and see what's changed and what's different. And then in the same way, you can look at it when you've treated a potential model organism like a mouse with different drugs and see how that potentially has changed the cells within the brain and how they're expressing different things. So, having this foundational map is really, I think, going to be such a critical part of how we're going to advance the sciences here using spatial biology.
So would a practical example be with something like, for instance, Alzheimer's or dementia, where these are progressive changes and certain things, there's amyloid plaques involved and all of that, trying to tease that out first to see what's the causal mechanism for these kinds of problems. And So
I think those are I think those are perfect examples. I mean, those are exactly the types of things that we want to understand better because, as you know, Stan, I mean, the field of neuroscience and neurodegenerative diseases, we've not been very successful in developing drugs that treat these type of diseases. And I think it still continues to be probably the highest rate of failure for drug development for drug companies, where we don't really understand sort of the causal mechanisms of why things are happening the way that they happen. And, having sort of the map that allows you to say, hey, here's what the normal state should look like. It allows us to start to trace back the different pathways and the different mechanisms that are involved when we look at disease that has progressed in these brains and potentially how those cells are interacting differently, how they've changed, what cells are there now and what weren't there before. So being able to do that type of analysis against these types of brain maps, I think ultimately is what really will help us most in terms of understanding how that disease progresses, how some of these neurodegenerative diseases are progressing. And then hopefully that does lead us to finding better treatments, better ways to look at what are the specific mechanisms that now we can potentially target with our drugs, and that will lead to improved conditions and outcomes for patients.
Will diagnostics be implicated here? Could you use this platform for diagnosing on an ongoing basis? I could imagine having some kind of analysis on an ongoing basis that says, gee, we've seen this movie before, and this movie takes you to this ending. And what can we do to change and have a happy ending?
I think with this type of technology, at least how it's being developed today, because it focuses on tissue samples, it's more likely to be kind of what I would consider front end in terms of discovery, in terms of identifying targets, confirming certain mechanisms of actions in model organisms. It's harder to apply this perhaps clinically in the neurosciences because typically we don't take brain tissues from from living patients, right? That's usually not good practice in most cases. But if we switch over to different fields such as oncology, where we are, as standard taking biopsies or tissue samples, this absolutely is an area where we can certainly foresee this being used in a diagnostic type setting, either as a prognostic tool, as a companion diagnostic, or as just for using as a diagnosis method. And on the protein side, I would say this is already advanced to some level where we're already starting to see the incorporation of spatial biology using it. Because what we've seen on the oncology side is that this complexity of how the tumor interacts with the immune system, it's very difficult to really identify what patients are going to respond best with certain types of immunotherapies, unless you really can see which cells are interacting with what types of other cells in those cases. And that complexity is what really lends itself very well to spatial biology, where you can really understand what is going on with this particular patient, what's going on with this particular patient's cancer. And then that allows us to say, whether or not a certain type of drug, which targets a certain pathway or a certain mechanism, is going to be successful or not in treating that patient.
Yeah, that's fantastic. I was just thinking when you were talking about brain samples, I would imagine that people studying CTE, there's been a lot of conversations about in the NFL about concussions and protocol and long-term effects of playing sports like an NFL football.
Yeah, I think certainly with potentially patients that have once they've passed and then their brains have been donated for some of this type of CTE research.
Yeah.
There's certainly opportunities then to kind of understand better what is happening when these concussions happen and how that's affecting the brain and what leads to that. So it's definitely the opportunity is there to really have that understanding of that biological progression or that disease progression in those particular organs.
So I'd like to pivot over a little bit. We've been talking about science, but I'm curious from a data science perspective, how do you actually merge the two types of genomic and histology information into something that's meaningful? Are you using AI and machine learning or deep learning, or how do you approach a problem of multimodal analysis?
Yeah, that's a great question. So from a technology standpoint, or from an informatics standpoint, a lot of what we've done in terms of the genomics information actually is very similar to what we would normally do with sequencing information or with single cell sequencing information. That sort of data is very similar to that type of data that we use today. What's added now, of course, is these spatial dimensions and the spatial information that we have from imaging. And there's a couple key things that have to happen in order for this to merge together. One very important component is this ability to do what's called cell segmentation. So when you take a picture of your sample, you just have the picture. The computer doesn't really know where is the actual cell located. And what we do in terms of trying to create the right images and using different staining approaches is to create boundaries around those cells so that those can be identified. And then there is a machine learning process that we would go through in order to be able to train the computer to identify that those areas that have been highlighted is actually representative of the boundary of the cell, right? And so by knowing that where the boundaries of the cell are, we can very specifically designate that as cell number X or cell letter X, right? And then all of the information located within that cell boundary area can then be assigned for that particular cell. So that is definitely one of the key components of being able to bring the information together so that we can use that biological unit of the cell as sort of a core component of any downstream analysis. But it has been kind of an area of ongoing development that digital pathology has worked in that field for many years, trying to improve cell segmentation and how to do it. And I think it's an area that's going to continue to advance and continue to get better in the future.
Yeah, so that leads me right to my next question. Thanks. Which is, where do you see you're a pioneer in these early phases of this technology? Where do you see it going over the next 3 to 5 years? What would you love to be able to say you'd be doing in the future with this technology? And
yeah, I think there is, we are such at an early stage in being able to bring this type of information to the research field. And I think step one is really make this as widely adopted as possible so people have access to the right types of data that they can use to advance their science and advance their research. One of the things that I didn't mention at the beginning is how spatial biology has kind of evolved over the last 5 or 10 years. And, the starting point for spatial biology really had been on the protein side. It really came out of the histology side with trying to do IHC, but doing more targets with IHC than we had before. And that allowed us to understand what are the different cells are located there. And so the protein side has actually been more, I'd say it's been around longer from that perspective. What we haven't had is this similar capability on the genomic side. We've never had data, or we've never had a commercial platform that could take this information from a tissue, understand, where the cells are, and then understand what genes are being expressed within those cells in the tissue. That type of information has never been available commercially. And so when we started Vizgen and launched the first platform in this space, this is the first time ever that now this data is accessible. So my hope is that over the next 2 or 3 years, that what we're going to see, and this is actually what we're seeing today, is a widespread adoption and quick utilization of trying to access this data, and then looking for what these clinical applications of where the clinical value is going to be downstream by using this scientific data.
Do you have any clients that you've served and are available, or stories that you can tell that are from your newsfeed that are public? I know we can't talk about proprietary clients under non-disclosure, but are there any that you can share about?
Yeah, I think one of the really exciting things that we've seen over the past year is going back to the brain again. And the Allen Institute, based in Seattle, has done a tremendous amount of research in brain research over the years, and they have been able to apply our platform to do exactly what we talked about, which is to map brains. And coming up, I think, at the Society for Neuroscience here next month, they'll be sharing a lot of that data there. And it is just amazing to see the amount of data that gets generated from, even one mouse brain, the number of cells that we're talking about, millions and millions of cells and tens of billions of RNA transcripts. Finding that information and then being able to apply that to get to the biological analysis of what actually is happening within the brain.
That's terrific. We've spent all this time talking about everything else but your company. So maybe you could share a little information about Vizgen and that you think people should know about your company.
Yeah, I'd love to. It's still a very young company. We were a spin out of Harvard University. This technology that I've been talking about for spatial genomics, it comes from a technology developed at Harvard called MERFISH. And that really has become the leading academic methodology for being able to do this high-plex, high-resolution spatial genomics. So when we started the company 3 years ago, the mission really was to be able to take that very elegant and powerful technology and turn it into true commercial product, commercial platform. And so within a very, very short period of time, within 2 years, we were able to launch the commercial platform, which is called MERSCOPE. So MERSCOPE has the ability to look at hundreds of genes in a panel, using a panel, soon to be up to 1,000 genes. And then by analyzing one sample, we can detect hundreds of thousands of cells and then potentially up to 1 billion different RNA molecules within those cells. And so it's been a really fantastic journey to see how fast that not only we were able to bring this to the market, but how fast customers out there in the research field have been wanting to, jump on this type of technology. Because I think in the genomics field, they've seen what's been happening sort of in the spatial world in general, but there hasn't been a tool to apply genomics into this field. And this has really been a huge breakthrough now to be able to get this available to these research scientists. So there's been tremendous amount of excitement. I'd say as a small company, we're doing our best to scale up as fast as we can so that we can meet the demand that's out there in the marketplace. But it's just been an incredible journey so far.
That's fantastic. I think my last question, your career has spanned a large area of life sciences, and you've grown up through the ranks from scientist all the way up to CEO of a company. Any advice that you could have for young scientists or, technologists who actually want to follow in your track and become, a successful executive like yourself?
Yeah, that's a great question. I think, what I've been able to do successfully during, my career is I've been lucky in the sense that I've had the opportunity to be in different parts of the organization and really understand what it takes to bring that part of the organization to be a great contributor for the company. So, just like life sciences, companies have different organs, right, that are contributing to the success of the company. And you've got the innovation side, you've got R&D, you've got operations, you've got your commercial side, but you've got quality, you've got regulatory. There are just many different areas that contribute. And what has helped me the most, really, I think, is having had experience in many of those different areas throughout my career path and really understanding the ways that they contribute to the overall success of the company has really helped me get to where I am today, so that I can apply that knowledge more broadly and help to bring up the company, hopefully in the right stages and the right steps to be successful.
It's so funny because your background sounds a lot like your current product, in that you have a neighborhood of different skills that you're putting all together and understanding the relationships with all those quote neighborhoods.
Yeah, it's very true. I try to apply the same framework, as much as I can to keep things simple in my head.
There you go. Listen, Terry, thank you so much for sharing your story and a little bit about Vizgen, and really giving us a little bit of a primer on spatial biology and spatial genomics. I really appreciate you coming and joining us.
Well, it's a real pleasure, Stan. Thank you very much for having me.
Take care now.
Thank you for listening to BioIT World's Trends from the Trenches podcast.